Sorry about that. We are back. All right, and so to finish off this part, let's see. The other things that you can do, other sort of common things, will be to look for a reduction in histopathology, which is disease you can see at the level of tissue. And so, for example, here would be what the virus does normally in liver cells. And so the liver cells are the nice sort of purple things, and these sort of pink expanses of just death and terrible stuff. That's what the virus is doing. And then here would be an example of after treatment. You'd have fewer and not as severe types of damage to the tissue. And so that would be something you would do in an animal. Something like a mouse would be common. Whatever animal is appropriate. So it has to be able to grow your virus, and often people will genetically modify the mouse to have the right receptor proteins to be able to grow the virus, because mice in general are cheap to use and are widely used, are decently well understood. You could try it in a horse, but you'd have to have a whole barn full of animals, and you need a decent number of animals. Sometimes you may need 15 or 20 in each of the treatment groups, treated and untreated, to get a statistically significant result and make sure that your drug actually works as you think. So, yeah, that is tricky stuff, and it helps to have a statistician involved, for sure. You can also look for markers in the blood of infection. So, for example, there would be markers that would show damage in the liver or damage to the heart or an inflammatory response, and these would come up on sort of a blood screen. And just like there are these blood screens that they can use on people when they go to the hospital, there are mouse versions of this that you can use to get a pretty good idea. Now, a mouse isn't a person, and that's why these things would be considered preclinical. And then for the actual clinical testing, you'd have to make a special batch of the drug using a thing called good manufacturing procedure, which is just very careful note-taking and quality control. And then you'd have to go through the stages of clinical trials. The phase 1, where you're just looking for what's the effect on healthy people. The phase 2, where you're looking for, all right, does this produce the right sort of response that looks as though it should actually have some effect against the thing we're trying to affect. So for like a vaccine, it would be looking to see if you make good antibodies, for example. And then phase 3, which is where you say, okay, we know that we have a drug that's not toxic. We know that we have a drug that produces something that looks like it should protect, or in whatever way. Does it actually protect? Do I get a difference in hospitalization, death, whatever? And when you're coming up with a study, you get to define what the protocols are, but you have to run it past the FDA or the equivalent organization, wherever it is you happen to be working. And they'll say, you've got to change your test, or, yeah, that looks like it constitutes a fair test. That's the idea. And then the last thing in the animals, you can look for physiological changes. Like a lot of animals will run a temperature, or here are some mice that are showing weight loss. And so you see weight loss drop down as the animal gets sick. Just because it doesn't eat as much, it just feels bad in some way. They don't move around as much. But eating can be a nice objective way to look at it. And you don't ever let the mice drop below something like 25% or 20% body weight loss. That's in a particular time. And again, you have to clear all these things with lots of different committees. You have to explain what is it that you're doing. You have to demonstrate that, is this a reasonable use of animals in research? Because you can't just do stuff in research, especially when it involves animals, and doubly especially when it involves people. So there we go. All right, let's finish out this section with a couple little things to think about. All right, so selectivity. This is going to be our big thing. How much compound does it take to have the desired effect versus how much compound does it take to basically start poisoning the cells, individuals, whatever it is you're treating. And so to do this, you figure out two numbers, and you've got the CC50, which is the cytotoxic concentration, 50%, the point at which you have killed half of the cells on the plate, just as a number to use. And then you'll get the EC50 is a common one, which is the 50% effective concentration. So if you were going to produce 100 viruses, this is the exact amount of drug where you only produce 50 viruses. And to do this, you do a standard curve. You do different amounts of drug, and then you can draw a line between them, and you can calculate where 50% would be and what concentration that would be. That's the idea. And so, you will use these two numbers to get a therapeutic index, which is normally the CC50, which is like the high amount it takes to kill a cell, divided by the EC50, which is the amount to actually have an effect that you want, which should be much lower than the CC50. And you basically see how big is the window, because you could theoretically put in drug up to near the point where you get cytotoxicity, and then, like I said, that gives you longer, because metabolism's going to start and filtration's going to start, and your drug's going to go down, 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 down. But if you start out higher, that means that maybe you only you have a longer period. So maybe you can treat once a day instead of 3 times a day. Or maybe you can treat 3 times a day instead of having a constant IV drip of the stuff to have the desired effect. And so having a good therapeutic index would be something like 100 or better. Usually the sort of knockout drugs are at least 1,000. Sometimes you'll see them up as high as almost 1,000,000. Yeah, and those are fantastic drugs with a big therapeutic index like that. There you go. All right, next. So I'll give you 2 examples of things that are not quite antivirals. And then all the rest of the lecture is going to be talking about things that are antivirals, and so I want to show you the difference. So the problem that both of these have is selectivity, essentially. And so this is a drug called cycloheximide. It looks like this. It's made by a bacterium, Streptomyces. They make a lot of antibiotics for us, these Streptomyces bacteria. It would not be considered a true antiviral, even though it will decrease the amount of virus present. So if you zoom all the way out, take a compound like bleach. Totally kills viruses, but totally kills cells. So the CC50 and the EC50 are very close to each other and are both at very low concentrations. And so that is not something that you want. Don't drink bleach, kids. Yeah. Stay in school. Don't drink bleach. Fine. Cycloheximide is kind of the same way. So cycloheximide affects the host. So it's actually a ribosome blocker. And what it does is it blocks ribosomes from getting together, which are called polysomes, when you have a bunch of ribosomes all translating the same messenger RNA. And it also blocks chain elongation. So as you're making the amino acid chain longer in your little ribosome, this gets in there and it just blocks that from happening. And so you just stop translation completely. So it completely affects the virus. You can use it at concentrations where the cell is just alive, but the virus is pretty well inhibited. But basically, when you do that, you are stopping all protein synthesis in the cell. And the cell can tolerate this for, I don't know, about 15 minutes, 20 minutes at a real push. And then after that, the cell dies pretty quickly. It's got to have a constant source of protein. So it is not a true antiviral because it hits the host just about as hard as it hits the virus. And so this is not something you'd ever want to use. This is a straight up poison and a pretty toxic one. And we would not want that. The other thing to look for, and actually let's jump ahead 2 slides, boom, boom, is resistance. So resistance is a hallmark of any true antiviral. And you'd say, wait a minute, I thought we don't want resistance, right? Yeah, which you kind of don't, but also resistance always happens because viruses are, remember this, little evolution machines. They evolve as a way of life, and they evolve faster and better than anything else out there. And so if your drug is specific in that it binds some part of the virus directly and inhibits the action of that part of the virus, then there will be some change that the virus can make that'll allow it to work just a little bit, grow just a little bit, and will make the antiviral not bind as well. It'll just change the shape of something so that the antiviral only kind of sticks and the virus still kind of works. Some kind of compromise like that. That's resistance, but the place where resistance happens tells you where your antiviral is binding. So if you have an antiviral that binds a polymerase, you will see resistance in modifications around the polymerase. Some part around the active site of the polymerase will change usually, and that'll tell you right where your antiviral is binding. So it's useful, and it's not all bad because of fitness. So remember our little fitness peak with our little virus up somewhere near the top on this? And it may be a big wide peak, or it may be a very narrow peak. We don't know until we see how the virus responds to evolutionary pressure. But what you're doing is you're forcing the virus to move away from wherever it is. So a virus will generally start out somewhere near the peak, and you're pushing it. And you'll find out if the slope is steep, the virus goes all the way down and it's dead. Or if the slope is kind of flat, you'll push the virus, but you'll only have a little effect on the virus right there. And so fitness is the ability of the virus to produce infectious progeny, which is basically more viruses that are capable of starting an infection. Just like it seems like it ought to be, just like it sounds. So it becomes a matter of how much of a cost does the virus have to pay? Because there's always a cost for resistance. The virus can change, but it's never a thing that the virus wants to change, because it would have already been changed that way in the optimal version. Yeah. And so if you have a drug that is effective against the nice optimal version of the virus, then you can push it. You can push it out of its comfort zone. And so the idea is, if you can just push it a little bit away from its comfort zone, even if it's on one of those big round peaks, let's say we push it to here with one drug, then you can come in with another drug, you're going to push it in a different direction. So let's say we're going to push it down to here. And if you come in with a third drug, you're going to push it again. And maybe you push it all the way down to there. And so whereas each drug would only push it a little bit ordinarily, used together, they can end up pushing it all the way down the hill to where the virus has a real hard time responding and replicating. Yeah. Because it can change, but if it makes too many changes, the virus is just dead. Yeah. So it's a big trade-off. And antivirals are never the complete answer. So antivirals will never work in the absence of an immune system, as far as I have ever seen. There's no antiviral that's 100% effective. What you're doing with an antiviral is you are buying time for the immune response, and you are limiting the size of the problem for the immune response to clean up. And so antivirals can suppress a virus, but an antiviral will generally not be able to cure a virus in any meaningful sense. And you hear that term get used a lot, but it's used by people who are non-specialists. And when somebody says, "Oh, I cured that virus," yeah, you know, and I know that they don't completely know what they're talking about, or at least they're not using those words in the precise way that one ought to when one understands a subject. Yes, shall. There we go. So, all right. So we talked about cyclohexamide. And cyclohexamide, you can get resistance, but resistance is at the level of the host. So the host can make ribosomes that aren't as good at binding cyclohexamide. And in that case, you'll get resistance of the virus, but also of the host. So it's the two are completely tied together. You are just stopping a common process. Yeah. Bad antiviral. Not really an antiviral. Yeah. Technically, I guess, but not one that I would ever want to use ever. Yeah, on anybody. Here's one. This is kind of an intermediate case. So hygromycin B is also made by a Saccharomyces, because of course it is. Yeah, there's a lot of these omycins are made by Saccharomyces. and it's going to inhibit synthesis by the ribosomes. And so once again, it's going to interfere with, prokaryote and eukaryote ribosomes. The neat thing here, though, is that this drug does have some effect on the virus rather than the host, because with certain viruses, when the virus goes into the cell, it disrupts host physiology just enough that now hygromycin, which normally kind of bounces off the cell and doesn't get in, it'll disrupt the membrane just enough, or the way that the cell brings things in, and so you'll end up with more hygromycin in the cell. So infected cells will get hit harder, and you do see this used. This is occasionally used in agriculture, hygromycin B. So selectivity is kind of at the level of the plasma membrane, and if you have a virus that changes the permeability of the virus membrane, then there's a chance that hygromycin will kind of work a little bit, but what you have to remember here is that there are different membrane permeabilities. Some cells make their living by sending things out and bringing things in all the times. Other cells just more or less sit there. They'll take in a little bit of food, but that's not really their main line of work. And so you want to make sure you're looking at whatever the most sensitive cells would be, and that's where you're going to see your effect. So hygromycin, I don't believe this is approved for human use for anything currently. Although if it is, whatever. Get in the comments, let me know. And so resistance is at the level of, some bacteria will encode a gene that actually breaks down the hygromycin, so that's neat. But eukaryotes don't do that, and viruses don't do that. You can get genetic resistance that we don't fully understand, but it's probably the virus changing in a way that does not affect the permeability of the cell quite as much. And this is probably something that the virus can change without any real loss of fitness, since this was just a side effect of virus replication, rather than something the virus has to do in order to make a living. So, all right, there we go. That's the theory of antivirals. And in the next lecture, we will get to some actual antivirals and how they work. All right, thanks very much.